Good morning, students. I am V. Vanitha, Assistant Professor of Botany. Now we are discuss about internal structure of monocot. So, you want to know about monocot stem. So, this image shows that the internal structure of monocot. So, the monocot stems in the vascular bundles are scattered throughout the ground tissues. So, like monocot roots and monocot stems are protected by an outer layer of the dermal tissues is called epidermis. So, the rest of the stem is made up of ground tissues and vascular tissues. Next, first the internal structure of monocot. So, the main represent the typical monocotyledons plant thin transverse section. So, of young monocot stems consist of first one epidermis. So, the epidermis is the outer layer of the stem. So, it consists of single layer of parenchyma cells, and the cells are compactly arranged. So, the cells is covered by the thick cuticle. So, cuticle layer is present, a few stomata are present, and the stem hairs are absent. Next, parts hypodermis. So, next layer of the monocot. So, the hypodermis of monocot is formed of the sclerenchyma cells. So, this is the stone cells. It lies below the epidermis and it is 2 to 3 layers. So, the tissues serve as a mechanical tissues. Next layer, ground tissues. So, it consists of thin-walled parenchyma cells with a intercellular spaces. So, the tissues extend from the below the hypodermis of the centers. So, it is not differentiated into the cortex, endodermis, pericycle, and pith. So, it is this character is a unique character of monocot stem. Next layer, vascular bundles. So, the vascular bundles are scattered, so all over the ground tissues. So, this also unique character of monocot stem. So, each vascular bundles is conjoint and collateral. So, the bundles are uniseriate and closed vascular bundles. So, the bundles are smaller in size towards the periphery and bigger in size. So, periphery means outer side of the stem. So, and bigger in size towards the center of the stem. So, this is the vascular bundle of monocot stems. So, the xylem always towards the inner surface and phloem towards the outer surface of the stem. So, main character is the cambium is absent in the vascular bundles. So, the vascular bundles are more in number, so towards the periphery, and less in number of towards the center. So, each vascular bundle is surrounded by a rounded sclerenchymatous bundle sheath. So, the bundle sheath is the unique character of monocot stem. So, there are xylem and phloem in each bundle. So, surrounded by xylem and phloem in each bundle. So, cambium is absent. So, the xylem is arranged in a Y-shaped manner. So, in the two arms of xylem and are occupied by the metaxylem vessels, and the tail is occupied by the smaller vessels. Smaller vessels forming the protoxylem. So, on the inner side of protoxylem is a water cavity is present. So, it is formed in a breaking of protoxylem vessels. So, the secondary growth is absent in monocotyledons because the cambium is absent of vascular bundles. The phloem lies above the xylem. So, the phloem is composed of sieve tubes, companion cells. So, in the monocotyledons, there is no phloem parenchyma, and the broken members of phloems is also found above the phloem is called protophlloem. phloem. So next, the diagnostic features are important or highlights of monocot stems. So the monocot epidermis is outer covering. It is made up of parenchyma cells, a cuticle present, stomata absent, the hypodermis present below the epidermis, and below the hypodermis interior to fill the ground tissues. The vascular bundles are scattered all over the ground tissues. The vascular bundles are conjoined, collateral and closed vascular bundles. So the vascular bundles are smaller towards the periphery and bigger towards the center. So each bundle is surrounded by the bundle sheath. Each bundle has a xylem and a phloem. So important point, in mecabium is absent. The xylem is arranged in the Y-shaped manner. So the phloem is composed of sieve tubes and companion cells. The protoploem is present above the phloem. So finally the secondary growth is absent because mecabium absent in monocot stem. Thank you.